Already we have discussed about glycolysis. The product of glycolysis are 2 pyruvic acids, 2 NADH2 and 2 ATPs. And second step, Krebs cycle, where those pyruvic acids enter in the Krebs cycle, yielding 4 NADH2, 1 FADH2, 1 GTP when 1 pyruvic acid entered in Krebs cycle. NADH2 and FADH2 are formed. NADH2 is having 3 ATPs of energy and FADH2 is having 2 ATPs of energy. But they yield ATPs only when they go to the inner membrane of mitochondria where elementary particles are there. In that step, NADH2 give 3 ATPs and FADH2 give 2 ATPs. And that final step is said to be terminal oxidation. Terminal oxidation even is also named as oxidative phosphorylation. Oxidative phosphorylation and is also named as ETS, that is electron transport system. Electron transport system. This final step is said to be terminal oxidation because oxygen takes part in final step and is said to be oxidative phosphorylation because here the formation of ATP from NADH2 and FADH2 in presence of oxygen. That's why it is said to be oxidative phosphorylation. And electron transport system. Here in the inner membrane of mitochondria, electron acceptors like cytochromes are there in the series through which electrons are flowing, hence the electron transport system. To study how this third step takes place, let us have the diagram. This is the inner membrane of mitochondria, magnified inner membrane, and this is outer membrane. In between them is a perimitochondrial space, and this is matrix of mitochondria. Already in Krebs cycle, NADH2 is formed. How this NADH2 yield 3 ATPs, that we have to see. This NADH2 undergoes dehydrogenation in presence of the enzyme NADH dehydrogenase. This is considered as complex 1. NADH2 undergoes dehydrogenation. That hydrogen enters into the inner membrane where FMN is there. What is FMN? That is flavin mononucleotide. That gets hydrogen, that gets H2. When FMN gets H2, it becomes reduced FMN, FMNH2. And remember, flavin mononucleotide is a hydrogen acceptor. They may ask one NEET question, which of the following is a hydrogen acceptor. Example for hydrogen acceptor. One NAD plus, FAD plus, FMN. The right answer is all. All these. NAD plus, FAD plus and FMN are hydrogen acceptors. See here, FMN receives hydrogen, gets reduced flavin mononucleotide, and that hydrogen undergoes splitting. Protons are pumped into perimitochondrial space and electrons move through the electron acceptors like ferrous sulfide through which in the membrane electrons flow, and now they are received by ubiquinone. UQ means ubiquinone, and this ubiquinone gets those two electrons and two protons from the matrix. Now it gets reduced into UQH2, reduced ubiquinone. And now once again here hydrogen gets splitting, protons are pumped here and electrons enter into the next electron acceptor, that is cytochrome B. Through the cytochrome B, electrons come to FES, then come to another ubiquinone. And here from this ubiquinone gets two electrons and two protons from the matrix. It gets reduced into reduced ubiquinone, and here hydrogen splits. Two more protons are pumped into perimitochondrial space. Now those two electrons enter into cytochrome C1 and cytochrome C. Finally, cytochrome A1, A3. From here, those two electrons are accepted by a terminal oxidant. That terminal oxidant is oxygen, atom of oxygen that receives those two electrons. That's why I have written half O2 here. And these two electrons and 2H plus and half O2 together forming H2O. This is the way of flow of electron. And here in this we can see totally 5 complexes. Here we can notice 4 complexes. And the first complex that consists of NADH dehydrogenase. And second complex second is here, where the protons from matrix are pumped in to the ubiquinone. This is complex second. And complex third consists of cytochrome B and a cytochrome C1. Cytochrome B to cytochrome C1 is a third complex. And fourth complex is cytochrome C oxidase containing cytochrome A1, A3. Cytochrome A1, A3. That is complex fourth. And this cytochrome C is a small protein which links complex third to complex fourth. Once in NEET, the question was asked, in electron transport system, complex third. What is complex third? Containing cytochrome B and cytochrome C1. Cytochrome B and cytochrome C1. From here to here is complex third. And complex fourth is enzyme cytochrome C containing enzyme cytochrome oxidase containing cytochrome A1, A3. This is fourth complex, and a small protein which connects complex third and complex fourth is cytochrome C as well. Once it was asked. And totally here we can see 4 complexes. In these 4 complexes there is a pumping of protons in 3 regions. Pumping of protons in 3 regions. And these 2 protons from this perimitochondrial space, they are pumped back into the matrix. That is in 5th complex. I did draw here, I am going to draw here as well. And when these 2 protons are pumped back into the matrix through the F0 F1 complex, where there is a formation of 1 ATP. When 2 protons are pumped back 1 ATP. Here we do have the pumping of 2 protons from matrix to perimitochondrial space in 3 regions, so that we can expect 3 ATP formation. 3 ATP formation. Let me draw that 5th complex also. This structure is a continued part of it. Here this is inner membrane and its outer membrane. This is perimitochondrial space. And already in these 4 complexes, in 3 regions, protons are pumped from a matrix to perimitochondrial space. In the 5th complex, what happens is 2 protons at a time can pumped back to matrix through F0 F1 complex. Already you know about F0 F1 complex. They are the 2 parts of ATP synthase. When these 2 protons are pumped back, the maximum energy is released, that is received by ADP and inorganic phosphate, leading to the formation of ATP. And here at the 3 regions, protons are pumped from matrix to perimitochondrial space, so that this we can expect 3 times the pumping back of protons from space to matrix through the F0 F1 complex. Hence the formation of 3 ATP from 1 NADH2 as well. This is the way from NADH2 3 ATPs are formed. And very important thing you must remember here is that there are totally 5 complexes. Which of the following is a first complex? Will be the question. What is the first complex? NADH dehydrogenase is a first complex. In this first complex, what happens? NADH2 undergoes dehydrogenation. That hydrogen is received by flavin mononucleotide. That becomes reduced flavin mononucleotide. That hydrogen splits and protons are pumped into perimitochondrial space, and electrons are moving through the inner membrane only through electron acceptors like ferrous sulfide, and these electrons come to ubiquinone, and this ubiquinone acts as a complex second, and this region only 2 protons from matrix will come into ubiquinone and 2 electrons from ferrous sulfide. Now it gets reduced into reduced ubiquinone. Once again, hydrogen splits, protons are pumped into the space and electrons enter into the next electron acceptor, cytochrome B, and ferrous sulfide and ubiquinone. In this place also, protons are pumped in, and this ubiquinone becomes reduced ubiquinone, and protons are pumped. This is the third region where protons are pumped into perimitochondrial space, and electrons will come to cytochrome C1. Remember, cytochrome B and from cytochrome B to cytochrome C1, this is the complex third. Cytochrome B to cytochrome C1 is a complex third, and this complex third is connected to cytochrome, connected to complex fourth. That complex fourth is having cytochrome C oxidase containing A1A3. Cytochrome C oxidase containing A1A3, and these two are linked by cytochrome C. And I have just mentioned which of the following connects complex third to complex fourth. That is cytochrome C, a small protein present towards the outer space of inner membrane that connects those two. And here we do notice the pumping of protons, 2 protons at 3 regions from matrix to space. And there is a fifth complex continued. At a time, 2 protons from the perimitochondrial space are pumped back into matrix through F0, F1 complex. Those are the 2 parts of ATP synthase. Here the energy is received by ADP and inorganic phosphate, leading to the formation of ATP. As there are 3 regions where 2 protons are pumped, and we expect 3 ATPs from NADH2. And oxygen is considered as terminal oxidant. They may make another question in the electron transport system, which is the final acceptor of electron or terminal oxidant. The terminal oxidant is oxygen that receives electron and also react with protons in matrix forming water. This is how ATPs are formed from NADH2, and how the ATPs are formed from FADH2. Already you know, from one FADH2, 2 ATPs are formed. That we can show here only. The formation of ATP from FADH2 doesn't have complex first. Instead of that, this complex second. Here only FADH2 undergoes dehydrogenation. Those 2 hydrogen atoms enter here only. H2 and reduced to ubiquinone. 2 protons are pumped in. The same thing is continued, but in only 2 regions protons are pumped, and those protons are pumped back, 2 ATPs are formed. So that for the formation of ATP from FADH2, complex 1 is not there. It starts from complex second, and that complex second is FADH dehydrogenase. This is complex second. That's all. Only at two regions protons are pumped in. Hence, two ATPs are formed from FADH2. It's about the terminal oxidation. The expected NEET questions here, they would ask which of the following are hydrogen acceptors, you can give, and you should know those complexes. Complex 1, that is NADH dehydrogenase. Complex 2, FADH dehydrogenase. Complex 3rd, cytochrome B to cytochrome C1. Complex 4th, cytochrome C oxidase containing cytochrome A1, A3. And which is the link between complex 3rd and 4th? A small protein named cytochrome C. And complex 5th, that contains F0, F1 complex. And from 1 NADH2, 3 ATPs. From 1 FADH2, 2 ATPs are formed. The terminal oxidant in this is oxygen, hence the name terminal oxidation. That's all.